Greetings, my name is Laura Giron, and today I'm gonna present you my investigation, which is called Long Intergenic Non-Coding RNA 1695 is upregulated in astrocytes under lipotoxic conditions and might promote angiogenesis in the brain. For a little background, a little context, obesity is a condition which is characterized by the excessive accumulation of saturated fatty acids in non-adipose tissues, leading to a phenomenon known as lipotoxicity. Previous studies have determined that these excessive concentrations can trigger inflammation, can increase the concentrations of reactive species of oxygen, and as well decrease the functionality of astrocytes, which in turn can contribute to the development and progression of neurodegenerative diseases. Taking into account the incidence of obesity and neurodegenerative diseases in the current century, the identification and characterization of the underlying mechanisms that could be or that might be present in these scenarios is encouraged. Taking also into account that this data could help increasing the knowledge and the comprehension, the understanding of these diseases and its connection to high-fat diet. In this study, then, we performed a transcriptome-wide analysis of astrocyte cultures exposed to toxic concentrations of palmitic acid, focusing on changes on the expression of long non-coding RNAs. Our study constitutes the first investigation of differentially expressed long non-coding RNAs in a lipotoxic setting in human astrocytes, allowing us then to advance in our understanding of the cellular mechanisms by which saturated fatty acids may promote neurodegeneration. For this purpose, we performed a RNA transcriptomic profiling, analyzing the differential expression of long non-coding RNAs in astrocytes under lipotoxic conditions. We obtained 111 long non-coding RNAs that were significantly expressed, of which long intergenic non-coding RNA 1695 was selected as a candidate for doing both the functional prediction and the enrichment analysis, taking into account primarily that this long-coding RNA hasn't been previously characterized. For this instance, we used LNC Deep program and the David platform for predicting the metabolic pathways and proteins from the interacting network in this long non-coding RNA, as well as the gene ontology terms associated for those proteins. As a result, adjusting the obtained data for biological and statistical significance, we selected 15 reactome terms, 20 biological processes, and 7 molecular function terms for the analysis of the 129 proteins that were potentially present according to our analysis in the interaction network of long non-coding intergenic RNA 1695 under this condition, under the lipotoxic conditions. The proteins were found to be related to processes such as the post-transcriptional gene silencing, lipotoxicity events, regulation of synaptic plasticity, immune response like neuroinflammation, and primarily to angiogenesis. um, as well as its inducers like interleukins and tumor necrosis factors, as well as their regulatory pathways such as Notch and WNT. Although angiogenesis is mostly related to um, to wound healing processes, it can become pathological under certain conditions that come with age and with high fat diets. For example, it has been previously linked to Alzheimer's disease by alteration in the cleavage of Notch1 and beta amyloid precursor protein, which is characteristic of Alzheimer's diseased brains. And also, it has been suggested that vascular impairment may lead to the pathogenesis of Alzheimer's disease. These results then indicate that excess saturated fatty acids deregulate angiogenesis in astrocytes, recalling what is observed in Alzheimer's disease and other pathologies with blood-brain barrier dysfunction, deformed capillaries, hypoperfusion, and neuroinflammation. Our findings then characterize the effects of long intergenic non-coding RNA 1695 in cultured human astrocytes under lipotoxic conditions and the stress by computational approaches, describing its putative interaction with 121 proteins associated with the stimulation of aberrant angiogenesis processes that lead and complement neurodegeneration, as well as with lipid metabolism, immune response, and additionally, it suggests that this long non-coding RNA could be an epigenetic regulator in the astrocyte's response to metabolic stress due to lipotoxicity, making it an ideal candidate for further analysis. This is then, as I said at the beginning, the first approach to analyze long non-coding RNA in human astrocytes under this kind of stress, these lipotoxic conditions, contributing then to a better understanding of the roles of long non-coding RNAs in neurodegenerative disorders, and providing directions for further research and experimental validation. Thank you very much.